Our next presenter, she is the professor, Department of Psychology, Faculty of Veterinary Science, Bangladesh Agricultural University, Manashi. And you can also add more information regarding
Respected Chair, thank you very much for your kind introduction, and also the co-chair and the learned audience. A very much good afternoon and welcome to my talk. আমরা এতক্ষণ অনেক ডিসিপ্লিনে অনেক কথাবার্তা শুনছিলাম, এখন একটু এনিমেল বা আমাদের যে প্রাণীরা রয়েছে তাদের সম্বন্ধে একটু আমি বেসিক্যালি আই গ্রাজুয়েটেড DVM, Doctor of Veterinary Medicine. I was a veterinarian, but now I'm focusing on my teaching and research actually, and specifically I'm a parasitologist. That means পরজীবী বিদ্যা। পরজীবী কথাটা নিশ্চয়ই আমরা অনেকেই জানি যে অন্যের উপর ডিপেন্ড করে বেঁচে থাকে। প্যারাসাইট যে হচ্ছে the parasite is the multicellular organism which depends on the host body, either human or animal, to complete his life cycle, and at the same time the parasite is doing very much harmful effect. এমন অনেক প্যারাসাইট আছে যেগুলো কিন্তু লাইফ থ্রেটেনিং, যদি প্রপার সময় ট্রিটমেন্ট না দেওয়া হয় তাহলে কিন্তু ফ্যাটাল মিন্স the animal or human must die. So parasites are not like we shouldn't neglect, and Bangladesh actually it is the museum of parasites, we can say. And আমাদের সবাই কিন্তু কম বেশি কৃমি, যে কৃমি বলে আমরা কিন্তু বাংলায় বলি ফিটা কৃমি, হচ্ছে চ্যাপ্টা কৃমি, এরকম অনেক কৃমি আছে। সো আমি আসলে কৃমিবিদ বলতে পারেন, কিন্তু আর এটা 4IR সাথে কিভাবে হতে পারে আমি একটু বলতে চাই। ওয়ান হেলথ শব্দটা we all may know about one health means human, animal and environment, and also the plant, of course. So all together is one health. When we will get a healthy human animals and also the healthy environment, then all together it will contribute to the, of course, the 4I event. So that's why let's try. So parasites, I am working on actually the different important parasites which are also important for Bangladesh as well. And one thing I want to say, there are many parasites which are zoonotic. Zoonotic means these parasites can transmit from human to animal and animal to human. So about 80% of the parasites are zoonotic. So we can, for human doctors and animal doctors, we are frequently working together because these organisms are zoonotic. Transmit can human to animal and animal to human. So I am working on actually my PhD work. I did my PhD in Hokkaido University, Japan. So my research was on leishmaniasis. The Bangla is কালাজ্বর। একসময় কালাজ্বর কিন্তু খুবই আমাদের যদিও এখন অনেক কমে গেছে, ভালো ট্রিটমেন্ট অ্যাভেলেবেল। তো কালাজ্বর কিন্তু এখন অনেকটাই কমে গিয়েছে, কিন্তু একসময় কিন্তু কালাজ্বর বা স্টিল ম্যালেরিয়া, আমাদের ম্যালেরিয়ার উপরও কাজ রয়েছে। সো এগুলো কিন্তু এখনো লাইফ থ্রেটেনিং এশিয়ান এবং মেবি আফ্রিকান কান্ট্রি গুলোতে। সো আমি আমার পিএইচডি, I will present a few part of my PhD work, and I will not, it is the second part of my PhD work. So I was searching that the reservoir host. Reservoir host means this host, the reservoir host means the host which can carry the organism, without development inside them. So a stray dog, we can see in Bangladesh we have many street dogs or stray dogs, which the dogs are usually didn't get any human care, even they are very close contact with the human. We see in the rural areas, even the urban areas, we see many street dogs are roaming here and there, but they didn't get any special human care, never. So these street dogs can be a source of zoonotic pathogens. As for example, rabies, we all know, jolatanko, we all know. So as for example, so these dogs can contribute for the spread of zoonotic parasites, and these dogs are the reservoir for the leishmania parasite, which is the causal agent of kalajar. So, and these stray dogs maintain the infection pressure of zoonotic helminth. Helminth means parasites. But this information on the parasite diversity in the dogs is very much few. So I will talk about something about which I use as a diagnostic marker is cell-free DNA. Actually, it was part of my PhD research. cell-free DNA means that DNA is liberated or liberated from cells and existing in the circulation. Circulation means blood circulation or serum or sputum, even urine. So this cell-free DNA, like you can see, this is the blood vessel, and inside the blood cells, and this DNA, cell-free DNA, is liberated from the cell and existing in the plasma or serum. So this cell-free DNA can be used for the detection of range of pathogens, including virus, bacteria, even parasites. So there are many previous publications who the scientists use this cell-free DNA for the diagnostic target for the parasitic infection, like Plasmodium, which is the causal agent of malaria, and Leishmania, I have already told, Trypanosoma and Schistosoma. All these are very much pathogenic organisms. So now I will say that we have analyzed the cell-free DNA using the high throughput sequencing. It has another name, next generation sequencing technique, NGS. In brief, we say NGS. So this technique is using in the already using in the clinical cancer research and non-invasive prenatal diagnosis. That means the fetal DNA can be found in the maternal plasma. And then even for the tumor researches, they are using this cell-free DNA. And also, of course, the identification of infectious microorganisms, bacteria, virus, even parasites, these organisms can be identified this technique. So my research objective was to evaluate this cell-free DNA as a potential diagnostic marker for the screening of parasite infections in dogs using this high throughput sequencing technology, or NGS, the Illumina platform. So the stray dogs, I have collected the blood from the stray dogs from actually the Kalajor endemic area, especially the Mymensingh. I have collected the blood samples from about 50 dogs. And then from the blood, the Buffy coat samples were separated. And then DNA was extracted from the Buffy coat samples. And then in the DNA, we have got the Leishmania organism by real-time PCR method. And this research have been already published in a very good journal. And after that, we have collected the blood in the FDA card also, and DNA was extracted from this blood. And then in another publication, we have found Babesia gibsoni and Anaplasma by PCR method. So in this research, we have used the plasma. Before I used the Buffy coat and FDA card blood, but this time for the cell-free DNA analysis, I have used the plasma, or in other words, serum, for the cell-free DNA analysis. So to carry out this research, at first the cell-free DNA was isolated from the plasma of 14 stray dogs, and by using the MACMEX isolation kit, and then the size and yield of the cell-free DNA were measured by using the this machine analyzer. And after that, the cell-free DNA was sequenced by using the Illumina MySeq platform. After that, the resulting data was analyzed by using this CLC Genomics Workbench specific software. This is the analytical workflow. After getting the MySeq reads, we have the sequence, we have got the DNA reads, we have normally call them the reads. So MySeq reads, after getting the MySeq reads, the low quality sequence were trimmed off. Low quality. After that, we have the rest of the reads, we have mapped the reads against the dog reference genome. Like, we don't need the dog genome, okay? We need the parasite genome. That's why we have mapped the sequence to the dog genome. And then the leftover genome sequences, we have did the blast search against the NCBI databases. And then the rest of the reads extracted having the best with parasite sequences. And the best quality sequences were selected mainly depending on the two criteria. First is the read length should not be less than 50 base pair. Another is the top 3 blast hits should be from the same taxonomic group of parasites. And then we have also searched for the repetitive 10-day motifs. So this is the result. So in result, at first, I want to say that the cell-free DNA concentration in the blood was ranged from 0.12 to 1.08 nanogram per microliter. That means very few. And the average size of the cell-free DNA, it is, and the cell-free DNA, the DNA outside the cell is always the short length. It will be not so big length. So around 200 base pairs. So in my case, I have found 160 base pair of the DNA. So this is the analyzer. This is the summary. This is summary of the sequencing result and blast search. So I will say in brief. So at first, about 98% of the reads were mapped to the dog reference genome. And among the blast hits, about 64% of the reads, we have found the blast hits. So this is the distribution of best hits in blast search. So we can say that we have found many kinds of hits like this is the dog hits, canis, bacteria, some viruses, and anaplasmatic. And I will focus on parasite. So after quality filtering, like previously I have mentioned that depend on two criteria. We have found that 150 hits were assigned to the parasite, which is my target. They are belonging to the 19 genera of genus. So this is the top 5 parasite genera species. We have found this analysis. This is the one kind of helminth, trematodes, protopolyestoma, diphylobacter. This is the fish tapeworm. This parasite can happen by eating the raw fish. Even babesia, which is also an important parasite for dog. And leishmania, which was our interest. 4 reads or 4 sequence reads were associated with leishmania species. So this table is showing the comparison between the cell-free DNA analysis and the previous PCR and real-time PCR results. So actually the BD means Bangladesh. So these are the dog ID. So we have found the 4 dogs were found positive both for cell-free DNA analysis and previous PCR analysis. These 4 dogs were common in both methods. And for leishmania detection, we have found 1 dog was common in cell-free DNA analysis and then real-time PCR analysis. We didn't found any commonly positive dogs in case of anaplasmatic. So this is the annotation. So we have searched for the annotation of the identified sequence, and then we have seen that for the babesia organism, we have about 7 regions. We have found the annotated sequences. And also for leishmania, we have found 3 regions of tandem repeat motifs, which are associated with leishmania. So in discussion, we can say that in case of babesia, which is also a very much pathogenic organism for dog, even livestock, there is a consistency between the result of cell-free DNA analysis and previous conventional PCR, which can suggest the usefulness of cell-free DNA as a biomarker for parasite infection. And in case of leishmania, the presence of cell-free DNA, leishmania cell-free DNA in the plasma might not assure the presence of parasites in blood circulating macrophages. But the cell-free DNA of tandem repeat sequences, which I have shown in the just previous slide, would be a suitable target for the molecular detection of leishmania. So in conclusion, we can say that the detection of cell-free DNA derived from expected and unexpected parasite in dogs this is for the first time. And cell-free DNA could be an efficient diagnostic biomarker for screening the parasitic infection in dogs using this modern technology, next generation sequencing techniques. But future research is needed to establish these parasite infections, and we should provide the precise data about the prevalence and impact of these diseases in dogs in Bangladesh. So I'm happy to share with you that this research topic have been published in the very much reputed journal Genomics in the 2019, and I have got the best publication award in Bangladesh Agricultural Universities Association. So I would like to acknowledge my PhD supervisors, Professor Kankatapura and another associate professor, and also my supervisor in Bangladesh, Professor Jahangir Alam from the Department of Parasite, and of course the leading program makes Japan for funding this research. Thank you very much for your kind and passionate hearing.
Thank you, Dr. Srinatha, for your nice presentation and good innovation. So, do you have a question?
Just have a question.
Yes. Yeah, that's good.
Yes. Thank you very much, Dr. Professor Dr. Shirin. Actually, you did a fantastic job. The zoonotic disease, zoonosis, it's a very, very, very dangerous thing. We are living together, our pets and animals and many other living beings. And somehow we may affect by our loving pets. We are taking dogs, cats in our home, but it should be disease-free. So this is very important. Actually, you are collecting data from the 50 dogs. They have no any measure. They are not treated any veterinary doctors or any management system. Livestock management system is not here. They are living everywhere in around our surroundings. We may affect it. Nobody knows. Thank you very much. Thank you.
Thank you, sir.
I am not asking any more questions because I will stay. Very good.
One query.
Oh, still one question. Thank you.
One query, but it is not actually আমি যেটা জানতে চাচ্ছিলাম যে 50টা ডগের যে ব্লাড নেওয়া হয়েছে এটা আসলে র্যান্ডমলি সিলেক্ট করা হয়েছে যেদিকে ভ্যালুসটা আসলে কিভাবে
ছিল কালাজ্বরের জীবাণু সেটা কোনো রিজার্ভার হোস্ট আছে কিনা আমি একটু বলেছিলাম যে রিজার্ভার হোস্ট মানে হচ্ছে যে যারা কিনা যে হোস্ট গুলো অর্গানিজম ক্যারি করে কিন্তু তারা কোনো সাইন সিমটম প্রকাশ করে না
যেটা জানতে চাচ্ছি যে এই যে সিলেকশনটা
আমি প্রথমে আমার টার্গেট ছিল কালাজ্বরের জীবাণুর রিজার্ভার সেই জন্য আমরা ময়মনসিংহ এর যে এন্ডেমিক এরিয়া ফুলবাড়িয়া ত্রিশাল ওই হালুয়াঘাট এইসব এলাকা আমরা কয়েকদিন গিয়ে র্যান্ডমলি এবং সব অল আর স্ট্রিট ডগস র্যান্ডমলি কালেকশন করছি কিন্তু আমরা ওই জিনিসটা মাথায় ছিল যে কালাজ্বরের জন্য এন্ডেমিক এরিয়া কোনগুলো সেভাবে আমার জাপান প্রফেসর তারা আসছিলেন
Thank you, Dr.